A very good evening to all of you. Today we are going to do the challenge of biochemistry. Why biochemistry is important? 16 questions in Neet PG belong to biochemistry. So if you score full out of full in 16 questions, 64 marks are yours. So don't neglect this subject. So let's begin with the first question. APO48 is a remnant of A. RNA editing, B. RNA interference, C. RNA splicing, D. DNA translocation. What happens in liver cell? ApoB100 gene gives rise to ApoB100 protein. But ApoB48 protein is made in intestinal cells from the gene encoding ApoB100 by the process of RNA editing, which is also known as differential RNA processing. It is also called as chemical modification of RNA. This is a post-transcriptional modification. Now, this RNA splicing is also a post-transcriptional modification, but this is removal of introns and not editing. So the correct answer here is A, RNA editing. Hepcidin decreases iron absorption by inhibiting A. Divalent metal transporter, B. Hep-histine, C. Ferroportin, D. Transferrin. See, this hepcidin regulates iron metabolism. It mainly regulates iron entry in circulation. By inhibiting ferroportin, hepcidin reduces the dietary iron absorption. Now, when there is a release of iron from macrophages, it is also reduced by ferroportin inhibition. So, increased hepcidin activity is responsible for reduced iron availability. So, increased hepcidin will lead to anemia. Decreased hepcidin will lead to iron overload. So, hepcidin synthesis and secretion by liver is controlled by iron stores within the macrophages. So, the correct answer here is ferroportin. Which of the following has highest glycemic index? Now, let us know what is glycemic index. This is the increase in blood glucose after the test dose of carbohydrate compared with that of an equivalent amount of glucose. This glycemic index is a value assigned to foods based on how slowly or how quickly those foods cause an increase in blood glucose level. The highest glycemic index is for glucose and galactose. Also, lactose, maltose, trehalose, isomaltose get converted to glucose. So they also have a high glycemic index. Glycemic index of fructose is less than that of glucose. Fructose and sugar alcohol have less glycemic index because they are not absorbed completely. Sucrose also has low glycemic index than glucose as it gets split to glucose and fructose. So the correct answer here is A, glucose. GLUT5 is a transporter for A. Galactose, B. Fructose, C. Mannose, D. Glucose. Yes, GLUT5 is a transporter for fructose. Please click the link in the description to join the Ultimate Neet PG Breakthrough Blueprint. This is a 3-day masterclass covering all the 19 subjects in a holistic manner, which will boost your revision to the next level. Also share this link with as many friends as possible so that maximum number of people are benefited. This masterclass is free for those who click the link in the description box today itself. Which of the following is insulin dependent? A. GLUT1, B. GLUT2, C. GLUT3, and D. GLUT4. Remember one thing, only GLUT4 is insulin dependent. Insulin regulates GLUT4 and this GLUT4 is present on membrane of peripheral tissue. So the correct answer here is D. Cause of HHH syndrome is hyperornithemia, hyperammonemia, homocystinuria is A. Deficiency of ornithine transcarbamylase, B. Defect in ornithine transporter, C. Deficiency of ornithine aminotransferase, D. All of them. It is a defect in ornithine transporter. The correct answer is defect in ornithine transporter leads to HHH syndrome, that is hyperornithemia, hyperammonemia, homocystinuria. This prevents ornithine transport into mitochondria, hence it in the cytoplasm, and its reduced ability to clear carbamyl phosphate. Which method is used to separate a mixture of lipids? A. Isoelectric focusing, B. Page, C. Chromatography, D. Electrophoresis. See, this gas liquid chromatography is used to separate mixture of lipids. So the correct answer here is chromatography. Wrinkled paper appearance of macrophage in bone marrow aspirate is seen in A. Tay-Sachs disease, B. Gaucher's disease, C. Niemann-Pick disease, D. leukodystrophy. Wrinkled paper or crumpled paper appearance of macrophage in bone marrow aspirate is seen in Gaucher's disease. So correct answer here is B. Gaucher's disease. In CRISP or Cas9 system, which repair mechanism is used for genome editing? A. Homologous repair, B. Non-homologous end repair, C. Mismatch repair, D. Nucleotide excision repair. It is non-homologous end repair. Rapport-Lubitz cycle shifts oxygen dissociation curve to right because of A. 2,3-DPG, B. 1,3-BPG, 3-phosphoglycerate, D. 1,6-fructose biphosphate. It is 2,3-DPG. Hot flushes are experienced as a result of A. Increased noradrenaline, B. Decreased estrogen, C. Increased noradrenaline and decreased estrogen. Increased. So correct answer here is increased noradrenaline and decreased estrogen. Restriction fragment length polymorphism is used for A. Analysis of DNA fragment, B. DNA estimation, C. Synthesis of nucleic acid, D. Detecting protein in a cell. It is analysis of DNA fragments. So correct answer here is A. Thermogenic food is which of the following? A. High protein diet, B. High carbohydrate diet, C. High fat diet. It is high protein diet. Ketone bodies are not utilized by A. Brain, B. RBC, C. Heart, D. Skin. So it is RBC. Enzyme deficient in hyperammonemia type 1 is A. Ornithine transcarbamylase, B. arginase, CPS1 arginosuccinate synthase. It is ornithine transcarbamylase. So correct answer is carbamyl phosphate synthase. Enzyme type of mutation. Type of mutation seen in sickle cell anemia is A. Insertion, B. Deletion, C. Point mutation, frame mutation. It is point mutation. Which replacement of amino acid does not alter its normal function? A. Glutamine replaced by arginine, B. Glutamine replaced by alanine, C. Glutamine replaced by glutamate, D. Glutamine replaced by aspartate. It is glutamine replaced by arginine. The clinical feature given below shows occurs due to deficiency of A. Tryptophan, B. Phenylalanine, C. Cysteine, D. Glutamine. It is due to tryptophan. Which of the following binds mRNA with ribosome in eukaryotes? A. Poly A tail, B. tRNA, S. methicillin. It is S. methicillin. Which of the following is analog property gradation? It is Conversion of pyruvate to oxaloacetate. Which of the following group of protein assist in folding of other proteins? Definitely it is chaperones. D. Myoglobin S which structure? A primary, secondary, tertiary, primary, secondary, tertiary, quaternary. It is primary, secondary, and tertiary. Best investigation for metabolic disorder is it is A. Western blot, tandem mass spectrometry, PCR, gel electrophoresis, tandem mass spectrometry. Separation of protein by their mass is done by gel filtration chromatography.